Welcome to this tutorial on CRISPR-Cas tools in Photo51. Today we will go over how to identify PAM sites and get the corresponding guide RNA for both traditional Cas systems and base editing systems. Start by opening your desired plasmid by double-clicking it or by selecting it and clicking Open in the top toolbar. Once the file is open, navigate to the CRISPR tab. You will see that the buttons are not available when looking at the plasmid in Map View. Switch to Sequence View to activate them. By default, CRISPR-Cas9 is selected. However, you can change the selection based on the editing system you're using. Go to the drop-down menu at the top left that now displays CRISPR-Cas9 and select a desired system. If you have developed your own unique system, you can also input this by going to Manage Systems at the bottom of the drop-down. This will open the Managing Modal box. Simply select Add New at the top left and input the parameters of your system. Once your gene editing system of choice has been selected, you can use the Show PAM Sites button at the top navigation bar to display all possible PAM sites in your sequence. You can either browse through the plasmid or use the navigator at the top. The navigator shows you the total number of detected PAM sites, which one you have currently selected, and lets you jump through each site one by one. If you click on a PAM site, it will display the corresponding guide RNA above the sequence and the cut site with a vertical line. If you would like to save a given guide RNA, you can first select the desired PAM site, then navigate to the Add Guide RNA button in the top toolbar. Click it. Simply give the guide a name and select Save. This will save the guide with the current file. If you want an overview of all guide attached to one plasmid, go to the navigation bar at the bottom and select the Guide RNAs button. It will show you the spacer sequence as well as the CRISPR-Cas system used. If you select the Info icon next to the spacer sequence, you will also get an overview of the CRISPR RNA with the spacer region highlighted in blue, the transactivating CRISPR RNA, and the full sgRNA sequence. Any of these can simply be copied by clicking the Copy icon next to the sequence. When working with base editors, Photo51 offers a couple unique features. You can easily filter the PAM sites for the desired codon conversion. Navigate to the codon conversion drop-down at the top bar. Select the drop-down, and you can now select if you want a specific type of amino acid or a stop codon either before the editing takes place or after the edit is done. For example, if you want to find a PAM site for a base editing system and you want to convert an arginine, then in the Before Edit column, I select the amino acid drop-down menu and select arginine. Now, only those sites that will edit an arginine are displayed on my plasmid. When clicking a PAM site for base editors, the nick site is displayed with a blue vertical line. The editing window is displayed in yellow, and the possible nucleotide conversions are highlighted in red. Again, once you have found the ideal PAM site, you can create and save the guide RNA by clicking the Add Guide RNA button in the top toolbar, giving it a name and hitting Save. You have now learned the basics of using CRISPR-Cas tools in Photo51. Thank you for watching, and check out our other Getting Started videos for more. Now it's time to keep calm and CRISPR on.